Welcome back to my lecture to the course on aspects of biochemical engineering. Till now, I was discussing that chemical reaction kinetics, then chemical reactor analysis, then enzymatic reaction kinetics, both by using free enzyme and the immobilized enzyme system. Now, today I am going to discuss the new topic, that is kinetics of substrate utilization, product formation and biomass production of microbial cells. So what I told you, you can remember that in which way this biochemical system is differ from chemical system. I told you the beauty of the biochemical system is that from a particular raw materials we can produce n number of products. As for example, if you look at glucose, we can convert it to ethanol, we can convert it to citric acid, we can convert to lactic acid, we can convert to acetic acid. So, different type of products we can form from one compound. But if you look at the chemical process, that as your product changes, your raw material changes. But here in the question come how it is possible in the biochemical system, how it is possible. This is possible due to the fact that that microorganism that plays very important role. The different microorganism has the different capability, because one, that's why I can give the example that yeast, as for example Saccharomyces cerevisiae, they can convert glucose to ethanol. Am I right? Now, if you look at this Aspergillus niger, it can convert glucose to citric acid. Now, Lactobacillus casei or Lactobacillus delbrueckii, it can convert this glucose to this lactic acid. So, that indicates that that your raw material is same, but you are getting the different type of products. So, this is very interesting. I hope that you will understand this thing. If you understand, then you can have better idea on the biochemical reaction kinetics. So, first thing that I want to tell here, that is that how the enzymatic system differ from the microbial system. Now, what we have in the enzymatic system? Enzymatic system, suppose just now I explained that glucose is converted to fructose with the help of glucose isomerase enzyme. Am I right? Now, so glucose, but if you use other than glucose, your glucose isomerase enzyme will not act. So, you require specific substrate, but the enzymes are very specific with respect to substrate. You cannot, it varies one particular specific substrate only it will attack and give the product. But in case of microbial system, it's totally different. When microbes they grow in a media, and in the media, like we are all human beings. For our survival, what we required carbon source, we required nitrogen source, we require minerals, we required vitamins. But similarly, microbes also, they required all this. They required carbon source, they required nitrogen source, they required minerals, they required the vitamin. So, totally that so what is the reaction mixture for the enzymatic system and reaction mixture for the microbial cell growth, that is totally different. So, here I want to emphasize that if you look at the difference, I told you the enzymes, basically they are globular protein, they are randomly folded, and in process they have developed some kind of active site, and this active site is responsible for the enzymatic reaction. And they are very specific with respect to substrate. They only at a particular temperature and pH, no ability to adapt to the changes condition or substrate sources, it does not have the ability. Only substrate is required to carry out it does not require anything else. You require only the substrate and proper pH and temperature, and cannot repair themselves or reproduce. So, this is the enzyme, that is the characteristics of the enzyme that we have. Now, if you look at the microbial system, the living organisms that carry out broad spectrum of biochemical reaction. I already mentioned that that different type of the same substrate can produce different type of products, and then acts on a variety of substrate. They can act on not only glucose, they can act different substrate they can use as a raw materials, and function optimum range of pH and temperature, can adapt to change the environmental condition and substrate. Let me tell you that this is the typical characteristics of the living system. One, what is the typical characteristics of the living system? The typical characteristics of living system is the acclimatization property. What is the acclimatization property? That means that suppose I can give the example that we are living in the tropical country. Now, when you go to the western country, they are very cold. So, immediately after going there, we will be having kind of setback, because we require some time to acclimatize with the environment. Then we can walk properly. Organism also like this. Organism, when you inoculate, it has a new environment for the organism. So, they required some time for this acclimatize. But slowly they will acclimatize. I can give a typical example. Now, astronaut, when they go to the planet, that moon and other places, then they in between the temperature shooted very high, maybe 50-60 degrees centigrade. Now, if we increase the temperature of the room, they suppose 45-50 degrees centigrade, we will run away, because we cannot, our body cannot tolerate. But our body can tolerate when we increase the temperature slowly, and our body will be adjusted at one time when we can adjust with. So, acclimatization property is a typical property of the living system, and another is they are very sensitive to the environment. The growth media is required comprising of carbon source, nitrogen source, vitamin and mineral. This I already mentioned. Can produce the and bounce back if it damages. So, that the reproduction, that is a typical characteristics. Now, here in the growth media, here I want to emphasize that this carbon source has 3 different purpose. What is that 3 different purpose? It is used as bodybuilding material, it is used as an energy source, it is used for the production of typical product. Now, nitrogen source mostly contribute for the cell mass formation. Now, vitamin and minerals, they mostly take part in the metabolic reaction, because if you look at our metabolic pathways, we require different enzymes, and all enzymes they required cofactor, and these cofactors mostly they are minerals or the vitamins. So, this is how they take part in this reaction. Now, let us discuss about the kinetics of the, because when we talk about the kinetics of the microbial cell growth, now the question comes that how we shall have to monitor the concentration of the cells. Am I right? Now, when we have the microbial cells might be of 2 types. One is your unicellular cell, and there is the multicellular cell, or filamentous type of cell. Now, in case of unicellular cell, the numbers is proportional to the mass. So, as for example, if we talk about the bacteria, suppose the bacteria they are mostly their unicellular cells. So, we know that what is the mass of one particular cell. So, if you even know that concentration of the cell may be grams per liter or milligram per liter, and if you know the mass of individual cells, if you divide by mass of individual cells, we will get the number of the cells. So, number and mass they will be interconvertible in case of unicellular cells. Unicellular cells, am I right? But in case of multicellular cell, it is not possible. You have to consider only the mass of the cells. Now, the substrate plus cells, how the reaction take place? Substrate plus cells, the extracellular product and cells. The cells, the reproduction is the cell is the factor. So, how you can write? This is substrate is consumed, the cell mass is this added, and this gives the product, and this more cell mass is produced. Now, net specific growth rate of the cell, how you can write? This is 1 by x dx by dt. So, this is x, x is when we express this mass per unit volume, am I right? So, but I told you in case of unicellular cell, mass and number they are interconvertible. So, if it is in case of unicellular cell, we can express this 1 by n dn by dt. So, this with respect to number we can do that. Now, now as you know that any kind of microbial population or any kind of living population, always there will be growth and there will be death. Actually, the net specific growth rate of the cells should be equal to the growth rate of the cells and death rate of the cells. That is the specific growth rate minus specific death rate, that should be equal to actual growth. Because one thing we should remember, that when we carry out any kind of microbial reaction, only living cell they participate in the reaction, then the dead cell will not participate in the reaction. So, then so that we shall have to take into consideration. What topic I have given here? This is the kinetics of modeling for the microbial growth system. So, we have 4 different type of models. One is structured model, unstructured model, unsegregated model and segregated model. So, the mathematical model, when you use any kind of cell growth, that we can have 4 different type of structure. Now, different type of models. Now, let us try to understand what do you mean by segregated model or the unsegregated model, structured model and unstructured model. Now, what is segregated model? Suppose segregated model means in our society we are so many peoples are here. Now, we find that some way that under no circumstances that one person is totally same as the other person. It is not possible. Even twin that we have, then twin children also there will be some kind of difference you will find. So, what is the actual case as that all the human beings, all the human population, their growth characteristics they differ from each other. Am I right? Now, if it is so, it is same is applicable to the microbial system. The microbes when grow, there are also different n number of cells are there. So, growth characteristics of the individual cells will be different. Now, if your mathematical model deals with the growth characteristics of the each cell individually, suppose in our society how we identify the different people with respect to their name. Now, in microbial population, if you put a marker 1, 2, 3, 4, 5, 6, 7, 8 and then we determine the growth characteristics of the individual. What is the growth characteristics of number 1 cell? What is the growth characteristics of number 2 cell? What is the growth characteristics of number 3 cell? And if if mathematical model deals with that, we call it segregated model. And what you call unsegregated model? When we assume the growth characteristics of all the cells 1, 2, 3, 4 they are same. Because this is the kind of idealist situation, because ideal situation means it is non-existent. The real situation is that is really that present. So, ideally when you make kind of ideal situation, then we assume the growth characteristics of all the cells will be same. So, then it will be unsegregated model. Now, what do you mean by structured model? Structured model means cell comprises of n number of biomolecules. They have RNA, DNA, protein and all these molecules they have. Now, when cell grow, this concentration of this RNA, DNA, protein and this may vary with respect to time. And if your model deals with rate of formation, the rate of formation of RNA, am I right? RNA. Then another model deals with the rate of formation of DNA. Another model may be with respect to some kind of protein, definite type of protein. So, then we call it structured model. But unstructured model is what? when we assume the rate of formation of RNA, rate of formation of DNA, rate of formation of different protein, they are not different from each other, they are producing at the same rate. So, that we call it unstructured model. So, what I want to tell that unstructured and unsegregated and unstructured model is the when we consider the one component solute, and this is called ideal model, ideal situation, and this is structured model is considered as the real situation that we have. Now, Monod equation, that is some kind of disimbalance with the Michaelis-Menten equation for enzymatic reaction, and this is actually on the basis of unstructured and unsegregated model. That is the ideal model, ideal condition, that mu equal to mu max S Ks plus S. And what is the S? S is called limiting substrate concentration, limiting substrate concentration. Am I right? Limiting substrate concentration. Now, what do you mean by limiting substrate concentration? It is very interesting. Suppose we talk about media, am I right? Because microorganism growth in a media, and media comprises of n number of components. It may contain nitrogen source, it can contain carbon source, it can contain minerals, it can contain vitamin. Now, suppose if you increase one particular component from 0 to infinity, keeping other component excess, then if you find that specific rate of growth is increases like this, and with respect to change of that particular component, then we call it growth limiting substrate. Now, it may be carbon source, it may be nitrogen source, it may be minerals, it may be vitamin, anything. But in case of enzymatic reaction, I told you substrate is very specific with respect to the enzyme. That cannot be changed. When you use glucose isomerase enzyme, then your substrate is only glucose. You get cannot be fructose, this can be only glucose. This is similar like this. So, mu is the specific growth rate of the cell, and mu max is this is the maximum that you can see, this is the maximum growth rate of the cell when it attains the plateau, and when mu by mu2 is there, then Ks will be equal to S. Ks is the saturation constant. Now, here I want to emphasize one thing, that what is the significance of the value of Ks. Now, if Ks value is low, that means you require low amount of substrate for getting a more amount of cell mass. If Ks is more, you require more amount of substrate for getting the same amount of cell mass. So, that is the significance of the Ks value. Now, this equation that if you look at that, when this, what is that equation that we have? We have mu equal to mu max S Ks plus S. Am I right? Now, when S tends to infinity, if you, this is the first situation, if it is very high, then we can neglect it. I mean, if we neglect it, then this Ss will cancel each other, and then mu will be tends to mu max. Am I right? This is exactly what we have written here. Now another, when you write this equation mu max S Ks plus S, now S will be finite when S is finite, because if S is finite, then I can say mu should be finite. Am I right? So, this you can add here, and another is that it does not explain when S is tends to 0. It does not explain what will happen when S tends to 0, and it does not take care the death of the cells, because the mu, that only that growth of the cells they have, but in the actual practice when cell grow, there is some death of the cells, and it does not take care the inhibition effect. There will be some substrate inhibition and product inhibition that is not considering in the Monod equation. Now, if you look at the different processes that we have, how you do the cultivation of the mode of cultivation of the microbial cells, we have three different type of processes. We have batch process, we have feed batch process, we have continuous process. Now, let me explain The fed batch I have already explained, you take the material at a time, let it react, after the reaction is over you take it out. Okay. In between you are not adding anything to the reactor and taking out anything from the reactor. But what is fed batch reactor? Fed batch reactor means you add the substrate in different slowly and slowly. So, initially let you take small volume of substrate, let the reaction take place, and then when substrate concentration decreases, again you add some substrate, so that you maintain the concentration below the inhibitor area slowly. So, there is the input of substrate and there is no output of substrate. So, that is what exactly we have shown here. There is the input of substrate, but there is no output of substrate. Now, when it comes at the total volume, then we stop the operation, we take it out. In continuous system is something different. I told you, whenever we operate any kind of continuous system, we first operate in a batch mode, let the reaction take place. When the rate of reaction is maximum, then we feed the substrate like this and continuously and take the product from this. So, here substrate is coming and product is coming out like this, and this is a continuous process. Now, whenever we handle any kind of microbial system, we should have a very good idea about the life cycle of the cells, because when suppose I want to handle a new microorganism and try to find out its capability for producing certain product, the first when you take a microorganism, it is totally black box for me, because black box means we don't know how this organism is growing. When it will be growing maximum, when it will go to the stationary phase, that we don't know. So, it is whenever we handle any kind of new microorganism, first our duty is to develop the life cycle, and during the life cycle, what are the things you have? You can have the lag phase, you can have the log phase, you can find out the stationary phase, and you can have the death phase. Now, every phase has its significance. What is lag phase? Lag phase is considered as the acclimatization phase. Acclimatization phase. So, organism requires some time to acclimatize with new environment. And what is the log phase? Log phase, or we call it exponential growth phase. This means here the organisms are very active. This is very active. Am I right? And this is the stationary phase. What is stationary phase? This is called starvation phase. What is starvation? Because you are not giving the sufficient amount of substrate to the organism. So, they starve for the survival. Here rate of death is equal to rate of growth of the cell, and death phase is the dying phase. Most of the organism they are dying at this phase. So, this is because what is the significance? Significance is that whenever we do any kind of inoculation, because when you work with any kind of microorganism, that we have to inoculate, and when you inoculate, you should be ensured your organism is very active. And when you should inoculate, this is considered mid log phase. This is considered mid log phase. Am I right? And this is the late log phase. Late log phase. So, your inoculation should be during these phases. That means, during this time, this inoculation, just to ensure that your organisms are very active at that time. Because if you inoculate the cell here, then every possibility that your population has lot of dead cells. So, your rate of reaction is a problem. So, this one has to keep it in mind. Now here what we have written, the lag phase, the cell adapt to the new environment and no or very little cell growth take place. So, mu net equal to approximately equal to 0. Log phase, the growth achieves its maximum. This we consider as the active phase, where mu net equal to mu max. And stationary phase is the growth this is the stationary phase, what we call starvation phase, due to starvation, where mu net equal to 0. Rate of growth is equal to rate of death of the cell. And in the death phase, this loses the viability and lives take place, mu net is less than 0. Now, if you do the cell mass balance across this reactor, particularly in a batch process, then how you can do that? The rate of input of the cell, because rate of input and output of the cell is equal to 0 in a batch process, because you are not putting anything in the reactor continuously taking out this is not you are doing anything. So, this will be equal to 0. So, rate of the growth of the cell, that rate of input, that rate of generation of the cells are generated. So, that will be mu g into x into v. So, we know that what is mu equal to 1 by x dx by dt, am I right? So, what is the dx by dt is the rate of growth of the cell, that is equal to mu into x. So, that what is dx by dt? X is mass per unit volume. So, this is the volume of the liquid, So, growth takes place along the whole volume. So, this is to be multiplied by v. The dx by dt into v, this we can write mu into x into v, am I right? Similarly, that here that accumulation in the batch system, the cell mass, that accumulation take place, what we can write dxv into dt, and cell mass death we can write kd into, kd is the specific death rate of the cell into x into v. So, this rate of growth, actual rate of growth, ultimately we can write mu max minus kd into x. This is the net growth rate, we can write like this. Now, how you can find out that that time required for the for changing the cell mass from x to x0 to x. So, this is the very simple way we can do that. We know mu equal to 1 by x. We know mu equal to 1 by x equal to dx by dt. So, this we can write in this form. Then we can write dx by, this is the integrate x by x0, the mu value, t value you can easily find out, that ln, we can write ln x by x0 divided by mu. This will is the time that required for a batch process. This is the batch process we can easily calculate. Now, in case of microbial population, two things we come across. One is called doubling time, another is generation time. Doubling time means the time required to double the cell population. And what is the generation time? Generation time means the time required for the cell division. Now, during the cell division, the one cell may divide into two cells. It may divide into 3 cells also. It may divide into 4 cells also. So since in case of generation time, during the cell division, whether one cell divided into 2, 3, 4 is there, so it is different. Naturally, not necessarily the doubling time is equal to generation time. Now, if your one cell divided into 2 cells, in that case generation time is equal to doubling time. So, here how we can calculate. Now, what equation we have written in the previously, T batch equal to what we can written, ln x by x0. No, but that x0 by mu, am I right? So, this is the doubling time means double the cell population. So, this x0 will cancel, then this will be ln 2 by mu. This is exactly this we have written the doubling time, this double the cell population. That what is the minimum doubling time? This is ln 2 by mu max. Mu max is the maximum specific growth rate of the cell. So, this is the minimum. So, suppose in a problem, if I give you that minimum doubling time of the cell is this, from that you can easily find out the mu max value. Now, if you want to find out that what is the generation time, then x0 will be converted to xn. Let us assume the xn is the we do not know that for part cell division how much cell is producing. let us assume for each cell division xn cell is produced, then if you, then your generation time will be ln xn by x0 by mu. Then we can calculate the generation time of the cells. Now, now in this there are different terminology we use. One is called specific growth rate of the cell, specific substrate consumption rate, specific product formation rate. I want to emphasize here that when we use the term specific, that is with respect to per unit cell mass concentration. But as for example, specific product formation rate of product formation per unit cell mass per cell mass per unit cell mass concentration, rate of substrate degradation, and the specific rate of substrate degradation, per unit cell mass concentration. Now, another term we come across, that is the yield coefficient. This is the growth yield, that is the dx by ds. The gram of cell produced per gram of substrate consumed, not added, consumed. So, that we should remember. Here also, when y p by s, this will be gram of product form per gram of substrate consumed. So, this we shall have to remember. And this equation that we can write dx by dt is this, that we can write it, and from this we can ds by dt, if we want to calculate this, we can write ds by dx, dx by dt, and dx by dt we can put the value mu into x, we can put, and this is equal to 1 by this we can write. This what we can write, this is equal to 1 by y x by s dx by dt. Dx by dt is the mu into x, and what is mu? Mu equal to mu max s into x. So, you can write it like this. Now, how you can find out the kinetic constant? We have shown in the enzymatic reaction 3 different plots we have. One is called Lineweaver-Buck plot, another is the Eddy-Hopsty plot, and Hensley plot. The similar way we can do that if we plot line 1 by mu versus 1 by s, we can find out from the intercepts 1 by mu max value. Slope, from this slope we can find out the value of Ks by mu max. If you put the value of mu max here, you will get the Ks. Similarly, Eddy-Hopsty plot we can find out the Ks and mu max value, and Hens and Wolf plot we can find out the Ks and mu max value. Now, what is the advantages and disadvantages of the batch process? Now, batch process, that is easy to set up and maintain, because if you look at our fermentation industry, mostly they are batch mode. As soon as the World Trade Organization, we have joint treaty, then all the, we have free trade in the different countries, then all the foreign company they enter into India, then our Indian company, they immediately they change over their technology to the advanced technology. Then batch process they are converted to the continuous process, because continuous process the productivity is much more as compared to the batch process. So, only the beauty of this process that is easy to set up and maintain, can use to study the life cycle of the cell, that is very important to handle the cells. Lower capital investment, reduce the risks of contamination or cell mutation of the as the growth period is short, and useful for the production of secondary metabolites. But the disadvantages is they cannot hold the log phase for longer period of time, because we have seen the life cycle. What is the life cycle? As the time passes on, one phase switches over to the other phase, because log phase switch over to the stationary phase. That is the major drawback with the batch system. So, if you want to, suppose you want to have more cell productivity in the system, so you should operate the system in a log phase for longer period of time. That is not possible in case of batch process. Lower productivity, that is one of the problem. Then required high downtime for cleaning and sterilization, and safety problem when filling and emptying cleaning the reactor, and batch to batch variability. The batch to batch productivity may vary. So, these are the several problems that we have with the batch process. So, in this lecture I try to tell the cell growth kinetics. Cell growth kinetics is very important. The reason is that a lot of biochemical industries are operated by using the different type of microorganism. The beauty of this system is that one particular substrate can produce n number of different type of products. The first question is that how you monitor the cell mass concentration. The cell mass can be monitored. If it is the unicellular cell, you can have either number of cells or the mass of the cells per unit volume. But in case of multicellular cell or the filamentous cells, we still have to consider mass of the cell per unit volume. Now, when we talk about the growth model, it may have maybe of 4 different types. One is segregated model, unsegregated model, structured model and unstructured model. And then monod equation is similar to the Michaelis-Menten equation, can be used for explaining the growth kinetics. Now, in the monod model has the different drawbacks, because it does not include the death of the cell, it does not include the inhibition of the cells, and here they have assumed that when substrate tends to infinity, the mu tends to mu max. When S is finite, mu is finite, but it does not explain what will happen when S tends to 0. Then I try to find out that what is the application of life cycle on the cell growth, because when you handle any kind of organism, life cycle plays very important role, because when you do the inoculation should be done in the log phase, mid log phase to late log phase, and then I try to find out how you can determine the growth kinetics is constant, as for example mu max and Ks, by using Lineweaver-Buck plot, Eadie-Hopsey plot, Hansen plot. And also finally, I discussed the advantage and disadvantages of the batch process. Thank you very much.